All right, let's go ahead and go into topic 2.4 on plasma membranes. At the very top of our handout, I pulled in the information from Collegeboard CED. You've got your enduring understanding, your learning objectives, and your essential knowledge. Now, I definitely think the essential knowledge is really useful for you guys to kind of keep referring back to, because this tells you exactly what you need to understand on this subtopic. So let's start with our topic. The cell has an outer envelope, which is known as the plasma membrane. The plasma membrane is a complex double-layered structure that's made up mostly of phospholipids and proteins. Now, it has an olive oil consistency. If we were looking at the membrane on its own, and if we broke down further in terms of how much of our phospholipids and our proteins, we would see 40% phospholipids and 60% proteins. So the plasma membrane is an important structure for us to understand because of all of the activities that are happening within the plasma membrane. Okay, so it is a very important role in the cell, and it's essentially regulating the movement of substances in and out of the cell, which is why there's a whole subtopic dedicated just to the plasma membrane, is because this is what controls what's going to come in to the cell and what's going to get out of the cell. Now, the plasma membrane is semi-permeable, and we've looked at the word semi-permeable before. It really means that there's only certain substances that are allowed to go through without help. Now, when we look at the plasma membrane, this is going to be small hydrophobic molecules such as oxygen, so O2 and CO2. Okay, when we say small hydrophobic molecules, this really will come back to the structure of our plasma membrane and how it is structured. So if you would look on here, we know that anything large or hydrophilic can only go through those special tunnels that we see embedded throughout the plasma membrane. The reason why it needs to be that way is if we look closely at the structure of our membrane. At the top of our plasma membrane, which sometimes is also referred to as phospholipid bilayer. Now, remember we looked at phospholipids back when we learned about our lipids. We have our phosphate group attached to the glycerol, and because this is our head, remember we looked at the structure of phospholipids back when we did our lipids, where we have our phosphate group with the glycerol creating our phospholipid head, which is hydrophilic. And that also tells you that it's polar. Remember that your hydrophilic components are usually going to be polar. At the bottom, we have our fatty acid tail, which we remember is hydrophobic, which is your non-polar. When we look at the structure of the plasma membrane, you're going to see that we have two layers of our phospholipids, and because we have those two layers of the phospholipids, that's usually why it's referred to as phospholipid bilayer. So there's two layers, and that's why they call it the phospholipid bilayer. So the membrane is structured with multiple phospholipids throughout, with your head facing the outside and the tails facing the inside. Okay, so the outside of our membrane is going to be hydrophilic, whereas the inside is hydrophobic. And that brings us back to what we talked about, that only certain substances can go through without help. Because the inside is hydrophobic, only small hydrophobic molecules can get through without the help. And anything that's large or hydrophilic needs special pumps because it doesn't want to go through that hydrophobic area. So hydrophilic so the heads are your hydrophilic area, and the hydrophobic area are your tails on the inside. The way that the layer is structured, we know that our hydrophilic heads are facing the outside. So other hydrophilic substances are not going to want to go past that into that hydrophobic area. And that polarity comes into play as well, because we know that our hydrophilic area is polar, our hydrophobic area is non-polar. Okay, so hydrophilic substances shouldn't go through the hydrophobic layer. This also ensures that within our plasma membrane, our phospholipids are never going to flip-flop to the other side, because that would involve the hydrophilic head having to cross through that hydrophobic area. Okay, so they're not going to be flip-flopping and really switching sides, because they're not going to want to have to go through the inner hydrophobic area. So the arrangement of our phospholipids and proteins give our model the name fluid mosaic model. The fluid in this name comes from the fact that both of our bilayers are flexible. So each layer of our phospholipids is flexible. That's why we say that it is fluid. The mosaic comes in because we have our membrane peppered with different proteins and carbohydrate chains embedded throughout of it. So that's why I would say mosaic. It's peppered. So there's several associated proteins on the plasma membrane because of the number of activities that are happening both in and on the plasma membrane. The first of which will be our peripheral proteins. So these are loosely bound to the inner and outer surface of the membrane. You can see them on the picture here. So they're loosely bound. The next are our integral proteins. These are known as amphipathic, which is similar to the plasma membrane. They have a hydrophilic and a hydrophobic region. Their hydrophilic region is going to extend out of the cell, whereas their hydrophobic region is interacting with the phospholipid tails. The interior. So these are firmly bound to the plasma membrane. Now, because your hydrophobic region, remember that it's interacting with the hydrophobic interior, you have to understand that it's nonpolar. Now, transmembrane proteins are integral proteins that are going to extend all the way through the membrane. Okay, so if it's extending all the way through, like we see on that picture, it's extending all the way through the membrane. That is our transmembrane integral protein. Adhesion proteins are going to be forming junctions between adjacent cells. Okay. Are receptor proteins. So these, for example, your hormones. Okay, so they're working very closely with hormones. They are docking sites for arrivals at the cell. Our transport proteins, the pumps that are using ATP to actively transport our solutes across the membrane. So, quick reminder about our solutes. These are those substance that is being dissolved. So solutes go into the solvent. Okay. Channel proteins are going to form channels that will selectively allow the passage of certain ions or molecules. Okay, so these are the channels within the membrane. Now, our glycoproteins are our cell surface markers. Okay, so they're exposed on the extracellular surface and they play a key role in cell recognition and adhesion. Your glycoproteins are really a carbohydrate side chain that is covalently bonded or attached to the surface of some proteins. And your glycoproteins are only going to be found on the outer surface of the plasma membrane. Now, an example of this would be your red blood cells and the recognition of ABO and RH blood types. Now, your glycolipids are similar to your glycoproteins. It is your carbohydrates that are covalently bonded to our lipids. Now, both our glycoproteins and our glycolipids, these are signaling molecules. Okay, these are signaling molecules that are going to really help distinguish one cell type from another. The last one we're going to look at are our cholesterol molecules. Our cholesterol molecules are embedded on the interior of our phospholipid bilayer, stabilize how fluid our membranes are in animal cells. Now let's take a look very quickly on the functions of some of these proteins that we just talked about. Okay, so one of the first functions that our proteins are helping our cells with is going to be transport. So this is the transport of molecules, electrons, and ions. So these transport proteins are really helping to carry those molecules, electrons, and ions through either channels, pumps, carriers, or even through the electron transport chain, which, as we remember, was our component that manufactures ATP. Another function is the enzymatic activity. For example, we have our adenylate cyclase, also known as CAMP. So this enzyme ends up synthesizing cyclase, or cyclic AMP, which is known as CAMP. Okay, so this will take ATP and end up converting that ATP into cyclic AMP, which is needed by your cells. Another function is signal transduction. So our binding sites on our proteins or protein receptors are going to fit the chemical messengers that are coming in, such as our hormones. Okay, so when those hormones come in, they bind to those binding sites, and based off the hormone that came in, the protein will change its shape and then pass that message forward into the cell. The next function we have is our cell-to-cell recognition. So some of our glycoproteins are in charge of identification. So they serve as identification flags that are then recognized by other cells. Okay. So this would be the function we're getting from our glycoproteins. Another function we can get from our proteins is cell-to-cell attachments. So remember when we talked about our junctions, so your gap junctions, your tight junctions, that cell-to-cell attachment is something that we're getting from our proteins. Another would be attachments to the cytoskeleton and the extracellular matrix. This helps our cells because it's going to help them maintain the cell shape. And it's also stabilizing our location. So the location of certain membrane parts or certain membrane proteins that we have within our membrane are being stabilized by those proteins.